So today we'll be looking at pinpointing the top questions and considerations about biosimilars. My name is William Wong, Associate Professor and Residency Program Director in the Department of Dermatology at Wake Forest School of Medicine.
And I'm Steve Feldman. I'm Dr. Huang's partner. I'm Professor of Dermatology here at Wake Forest.
In regards to the learning objectives for this session, 1, we want to be able to define biosimilars, extrapolate the indications and interchangeability on the basis of FDA definitions and guidance. 2, explain the similarities and differences between biosimilars and originator or reference biologics, as well as the differences between the terms biosimilar and generic. 3, evaluate key aspects of the current FDA guidance that inform the healthcare provider's knowledge of the biosimilar approval process, manufacturing, and naming, as well as state regulations surrounding substitution practices. And 4, apply foundational knowledge on biosimilars to clinical situations relevant to dermatology practice.
Biologics have been revolutionary for our patients, and now we're starting to see biosimilars, and the number of biosimilars that are already approved is growing.
In regards to a quick review of the core concepts, number 1, we need to understand that biologics, including biosimilars, cannot be made generic due to the complexity of the molecule and variations over time. Biosimilars are biologics. Now, to understand this, take a look at this chart. On the left, we see aspirin with a molecular weight of 180. This is a small, low molecular weight molecule that has a simple, well-defined structure. Its manufacturing is reproducible through chemical reactions, and exact identical copies can be made. It is a molecule that has been completely characterized and is relatively stable, and for the most part, it is non-immunogenic. Now looking on the right-hand side with biologics. A typical monoclonal antibody has a molecular weight of 150,000. Due to its complex, heterogeneous structure, it is nearly impossible to fully characterize the molecular composition. Biologics are produced in living cells or organisms, and it is nearly impossible to ensure identical copies. In general, biologics are unstable and sensitive to external conditions, and they are immunogenic.
I think this slide really speaks to the foundation of what we hope to get across in this presentation. Biologics are such large, complex proteins. They're a mixture of different proteins and glycosylation products that nobody can duplicate them, not even the Innovator company that designed them. So from batch to batch, there's going to be variation, and this is critical for understanding biosimilars. If you look at the biologics that are available, they've gone through multiple changes in their production over the time they've been available. So with each change in productions, there's going to be minor variations in the products. Now, the products are going to be very similar from batch to batch, but they aren't identical. Identical copies of biologics simply can't be made. So with generics, yeah, you can make the original small molecule the same from batch to batch, and the small molecule generics of it can be identical. But the reference biologics vary over time. They're a mixture of things. They're so complex, they can't be duplicated. Biosimilars are very similar to the reference biologic, but they're not going to be an identical product.
So to highlight this, the second core concept is that a biosimilar is a biological product that is highly similar, but not exactly the same, to a U.S. licensed reference biological product for which there are no clinically meaningful differences in safety, purity, or potency of the product. Now, the comparability exercise used to demonstrate that a biosimilar is highly similar to a reference biologic is scientific, robust, and regulated.
The way I like to think of this is that the biologics that we've had, the Innovator products, have been absolutely revolutionary for our patients, even though they aren't the same from batch to batch. The fact that there might be some slight variation from batch to batch or between a biologic and a biosimilar is of no meaning to me, because These are revolutionary drugs in terms of their efficacy, safety, whether there are these minor variations or not. Well, let's think about this with a specific case. Consider a 49-year-old who has long-standing psoriasis. This is a horrible disease. 20% of the body is affected. It's affecting every aspect of their quality of life, how they feel about themselves, how they think other people feel about them. They've gone through multiple treatments already, including topicals, phototherapy, and methotrexate. Now they're thinking about going on Adalimumab. Considering the treatment with Adalimumab, there's a variety of options. There's the Innovator product, and there's a couple biosimilars. They're all dosed identically with a standard 80 milligrams initial dose followed by 40 milligrams a week later, and then 40 milligrams every other week after that.
Now, in looking at the FDA process for biosimilar approval, we want to make sure that this is rigorous, and we want to make sure that we have confidence in this process. Now, on the left, you'll see the process of creating, again, exact copies of generics. Again, these are small molecules, and they are approved through the Food, Drug, and Cosmetics Act. The initial small molecule has to be approved through a new drug application. They have to fully report the safety and efficacy investigations, and there are two pathways for approval based on the right reference. For a generic of that small molecule to be approved, it goes through an abbreviated new drug application. The molecule itself is identical to an already approved product, and there are no safety and efficacy data required, only bioequivalence. On the right-hand side, you'll see the approval for a biosimilar. Again, biosimilars are similar, but not exactly the same as the Innovator product, and this is actually approved through the Public Health Service Act. Initially, biologics have to go through the Biologics License application. They have to fully report the safety and efficacy investigations, and the applicant has right of reference to essential investigations. For a biosimilar, they go through the Biosimilar Biologics applications. They have to prove that their product is highly similar to the Innovator product. In addition, they have to show additional data showing absence of clinically meaningful differences. Interchangeable biologics are approved under the biosimilars pathway, but must meet higher standards. Now, in looking at the evidence that is used to approve a biosimilar, we look at the totality of the evidence. We look at the analytical studies, the animal studies, the clinical PK and PD studies, the clinical immunogenicity assessments, plus we add additional clinical studies. The ultimate goal of a biosimilar development program is to demonstrate biosimilarity between the proposed product and a reference product, not to independently establish the safety and efficacy of the proposed product.
Keep in mind that the current batch of the Innovator product is not identical to the original product that underwent studies. It's still a wonderful revolutionary drug, but we have very little data to prove to us that the current batch is not different from the originator product. In contrast, look how much data and studies that Dr. Huang has already described as to what gets done to evaluate a biosimilar. In the biosimilar comparability exercise, you have a deep base of analytical information showing that the molecule, the drug, the biosimilar is very, very similar to the reference product. On top of that, you have clinical pharmacologic studies, and in addition, maybe even a clinical trial showing that the drug works the same way that the Innovator product does. Well, let's go back to our case of our 49-year-old with long-standing psoriasis, considering treatment with Adalimumab, or potentially one of two biosimilars that are also available. The data that we have to support that the biosimilar is similar to the Innovator product includes this whole array of research that's done. The primary structure of the protein, how that protein is arranged in three-dimensional space, whether they're aggregates, the charge on the molecule, and what sugars are on the molecule as well. All of that has to be very, very similar to the originator molecule. So similar that you wouldn't expect any clinically meaningful difference. In addition to that, In addition to showing that it's similar to the reference product in those ways, you have to show that it has similar potency in various binding assays and in pharmacologic studies that look at the similarity. The slides on the pharmacokinetics of the drug I find to be one of the most convincing aspects of similarity of biosimilars to reference products. Look, first you test whether the biosimilar binds the target the same, and then you look to see if it stays in the blood, if the biosimilar stays in the blood the same. Here you see the pharmacoequivalency of a biosimilar and the reference product, and they basically line up right on top of each other.
Now, in regards to looking at some of the confirmatory clinical studies, here we see equivalent efficacy in the indication of rheumatoid arthritis between the Innovator Adalimumab and one of the biosimilars. As you can see over time, these two curves are almost exactly the same in the percentage of patients achieving ACR 20. For dermatology, there's additional clinical confirmatory studies looking at the equivalent efficacy of plaque psoriasis. And again, over time from baseline, 4 weeks, 8 weeks, 12 weeks, and 16 weeks, you see the mean number of patients having PASI % improvement is almost nearly identical. There are other studies that show similar efficacy and safety of biosimilar adalimumab and the reference product in patients with rheumatoid arthritis, with the number of adverse events being almost nearly identical. Looking at the safety, the immunogenicity of the Innovator and biosimilar adalimumab across both the indications previously discussed, including rheumatoid arthritis and plaque psoriasis. We see the binding antibody positivity and the neutralizing antibody positivity nearly identical between the two products in both of the indications. In addition, the development of anti-drug antibodies is very similar between the reference molecule and the biosimilar adalimumab in both the biosimilar, the US-approved adalimumab, and the EU-approved Humira.
I think when we're looking at biosimilars, we know that they bind the target molecule the same. We know that the pharmacokinetics is the same. One of the things people worry about, I think, is that the minor differences between the biosimilar and the Innovator is the potential for development of immunogenicity. Of course, we have that same potential issue with batch-to-batch variation within the Innovator, and if you're not worried about that, then I'm not sure that we should be any more worried about the biosimilar. But the data seem to support that the risks of anti-drug antibodies are not greater with the biosimilar than with the reference product. Well, let's go back to the case. Again, we're considering treatment with adalimumab versus the other options, a biosimilar version of adalimumab. Dr. Wang, what would you say the key take-home points are at this point?
I think one, in comparing generic medications versus a biosimilar medication, that there is a much more rigorous approval process for a biosimilar. In addition, we have additional data and large trials to support both similar efficacy and also safety for these patients. Now, I would understand the audience being a little bit hesitant in starting a biosimilar because this is a relatively new concept. But I think you bring up great information that from batch to batch from the Innovator product, that there are inherent differences. Again, these are large, complex molecules that are not completely understood, and that from batch to batch, there can be key differences. And we see in these studies that in biosimilars, they have a very similar efficacy and similar safety across different disease states.
I think it's really easy when you're starting a patient on a new drug that, you could almost flip a coin or do whatever the payer said to do with regard to starting on a reference product or its biosimilar. But let's consider the case now of when you have a patient who's doing well on their current Innovator product and they're wondering whether to switch. Let's consider a 53-year-old who's failed non-biologic DMARDs and being treated with Remicade. She's doing well on it for 4 years now. At her last appointment, and this is not surprising, she gets a letter from her insurer saying, you know what, your copay will be less if you switch to the biosimilar. The FDA has already approved biosimilars for infliximab. They are already on the market, and so this is a practical question that's going to come up.
Now, in looking at the efficacy of the biosimilar and the Innovator infliximab, what we see in this table is that they have equivalent efficacy in patients with rheumatoid arthritis. Now, in terms of the treatment difference, you could almost argue that the biosimilar shows slightly higher efficacy than the Innovator. But in looking at the overall big picture, they're actually quite the same. Now, in looking at the patients that responded and received an ACR 20, again, on the left, you see the two tables look very similar, and the curve is almost exactly the same. And these are looking at equivalent efficacy in patients with rheumatoid arthritis. Now, there are several biosimilar studies that may be designed to address considerations for switching treatments, the question that is brought up in the case. This defines the term interchangeability. On the top is an example of a transition study, where a patient is on a reference drug and another arm is on a biosimilar, and then at some point in time, all the patients are on the biosimilar. In the middle, we see an example of a single switch study in which a patient is on a reference drug and then gets switched to a biosimilar. The last table you can see is a multiple switch study where patients in both arms are constantly being switched back and forth, and during this period of time, efficacy and safety are being monitored. Now, in the NOR-SWITCH study, which is a non-inferiority study, this was a phase 4 multi-indication prospective non-medical switch study in Norway by the Norwegian government. They looked at adults with a wide range of inflammatory conditions, and it was a 52-week randomized double-blind non-inferiority study, which you can see the design below. And essentially what they saw was that there was really no difference across a wide range of worsening once patients were switched from one product to the next. They also showed no difference in the development of anti-drug antibodies. And as you can see in this table, those in the originator group or infliximab, 7% developed anti-drug antibodies, whereas in the biosimilar, it was 8%.
In this study, patients were randomized to either the reference infliximab or to a biosimilar, and they were followed through week 78. And you can see the response to the Innovator and to the biosimilar are essentially completely identical. The issue we have in this case is that the patient's already doing well on the reference product, and psychologically, the patient probably is not going to want to rock the boat in any way, despite all this data showing that the biosimilar is actually very similar to the reference product in every clinically meaningful way. But here's the rub. The patient, even if she stays on the reference product, she's switching. Every batch of drugs she gets may be different than the previous batch, even if she's staying with the reference product, because the reference product is so big, so complicated that nobody, not even the Innovator company, can perfectly duplicate it from batch to batch. So there's always going to be some uncertainty. Here, if she switches to a biosimilar, there's a massive data showing that you can reasonably expect that biosimilar product to work the same way that the Innovator does. This slide shows further data on transition from reference infliximab to the SB2 biosimilar form of infliximab, and you can see that the side effect profile is really very same across the different products, and if anything, there might be a slightly lower risk of developing antibody with the biosimilar than with the infliximab. Dr. Wong, did you have any other thoughts about this idea of switching a patient who's doing well on a reference biologic and who thinks that the reference biologic must be the same from batch to batch because the name doesn't change?
So I think this gets to the heart of the reason for this educational module, is to understand what truly is a biosimilar and how that is different from a biologic. For example, if you prescribed a patient Dazoin and they ended up getting generic Designide, you wouldn't have second thoughts about it. But we know that from batch to batch, the Innovator product can change due to its complex nature. So if a patient is switched from a biologic to a biosimilar, and we have studies to support similar efficacy and similar safety, we should ensure the patient that this is a safe switch.
You know, in this case, the insurer didn't say you have to switch. They just said you get a lower copay if you switch. And I think that's a very reasonable approach that gives patients an incentive, lets them choose to some extent. And given the high quality of the data showing the similarity, they might reasonably choose to take the lower cost product. Well, let's consider another situation. Here we have our 53-year-old patient with psoriasis, and again has failed the DMARDs, was treated with the branded infliximab at the standard dose, and was doing great for 3 years, but now the drug stops working. The symptoms are poorly controlled. The disease is coming back. The blood work shows that the infliximab trough levels are low and anti-drug levels are high. Is this a good time to switch to the biosimilar? Anti-drug antibodies between a biosimilar and reference product are going to cross-react. The biosimilar is basically like, in my way of thinking, another batch of the reference product. They're just so similar. So that if the reason the drug's not working, if the branded infliximab is not working because of an antibody against the infliximab, switching to the biosimilar is not going to help at all, because it is basically like giving the original product. The antibody is likely to bind to it, and it's likely to make the drug level of the biosimilar disappear just as quickly as it does the branded product. Any thoughts on this case?
So I think in terms of the last two cases that we reviewed, patients are considering a switch for very different reasons. In the first case, the patient was considering a switch because they got a letter from their insurance company saying that her copay would be less if she switches to a biosimilar. She is otherwise doing very well on the molecule itself, and for here, it may be a reasonable alternative for her to switch to a biosimilar. In the second case, it's very similar, except the patient is not doing well on the biologic medication and has evidence of low drug levels and high anti-drug antibodies. And as you noted, the antibodies will cross-react between the biosimilar and the Innovator product, and for here, a switch is not warranted. Now, these scenarios will continue to come up and it will invariably occur in your own day-to-day practice if you prescribe biologics. The key to all this is to ensure the patient's safety and to ensure consistent patient outcomes when thinking about making a switch.
You know, I am totally comfortable with the idea of biosimilars. To me, they're just like the Innovator product. But if you have a patient who's doing well on one, there is some non-zero probability that at any moment that drug may stop working for them. And if you switch them to a biosimilar and you catch them when the drug is going to stop working, it may have nothing to do with the fact that it's a biosimilar. This class of drugs was going to stop working for them. Then the switch to the biosimilar is going to get the blame. So, you know, psychologically and medical legally, you know, I really like the approach where the insurer says to the patient, listen, we'll give you a lower copay, you decide. And so that it makes it the patient's decision and not something I will get the blame for, because, you know, even though I don't think that switching to a biosimilar would cause any problems happen. Extrapolation refers to the idea that a biosimilar might be tested in one disease, but then gets approved for all kinds of diseases, whereas the original Innovator product, it was tested in all those other conditions. Would we feel comfortable using biosimilars for diseases in which they weren't tested? And my answer is, of course, yes. When the FDA approves a biosimilar tested for one indication, you can expect that it's probably going to get approved for all the indications of the original product, in part because you've already shown that the structure and the function of the molecule and its pharmacokinetics, all of those things are similar. And then they try to test in the disease that might be most sensitive for detecting differences. If they don't detect a difference there, you've pretty much put every nail in it that you possibly could, showing that extrapolation is reasonable. But here's my biggest reason. Biologics, they're revolutionary drugs. They're wonderful, but they vary from batch to batch. And the current batch has not been tested, as far as I'm aware, in any disease. So if a biosimilar is brought to market and has been tested in one disease, I actually have more information about its effectiveness than I do for the current batch of the Innovator product. I understand, Dr. Wong, that biosimilar etanercept was approved for patients with psoriasis. Do you feel like there's, the data to make you feel comfortable using it in psoriasis, using it in other indications?
Yes. So as we're looking at this slide, we're comparing the etanercept biosimilar and etanercept Innovator in healthy subjects. And in this particular slide, we're looking at the mean serum concentration and the PK equivalents, which again, these two curves are nearly identical, as close as you can get to being identical to one another. Now, the Egality study also looked at the biosimilar versus the originator product in patients with plaque-type psoriasis and looked at various endpoints. As you can see throughout the particular trial, and we have data all the way up through week 52. Now, in comparing the treatment response at 12 weeks in patients with plaque-type psoriasis, we can see an adjusted PASI 75 nearly identical between the biosimilar etanercept and the etanercept Innovator. Now, the efficacy outcomes were again up to week 52, and what we saw was equivalent efficacy and a lack of difference between the treatments between the two products. Switching treatments also did not have a negative effect on efficacy. Again, looking at safety data specifically, there were no significant differences in treatment emergent adverse events or anti-drug antibody formation between the etanercept biosimilar and its Innovator in patients with psoriasis. So I would say, to answer your question, Steve, yes, there is a lot of data to support switching a patient from Innovator and etanercept to the biosimilar based on the studies that we have currently available to us.
I look at those pharmacokinetic studies and see the lines on top of each other, and I think I would have expected that if you took two sets of patients, put them both on, say, the reference etanercept, you might see more difference in those lines than you do just giving people the etanercept reference product versus its biosimilar. It's really striking.
Now, as we talked about before, extrapolation is a novel concept. In addition, the concept of interchangeable is an FDA designation. Now, we talked about some of the standards that are required for establishing biosimilarity. However, the idea of interchangeability requires a different set of data standards. This includes dedicated switching studies, which we reviewed earlier, in addition to post-marketing monitoring. Again, a product with an interchangeable designation may be substituted without intervention of a prescribing provider. Now, state by state, state substitution laws will impact this practice. In addition, any biological product under consideration for substitution must first be approved by the FDA as quote-unquote interchangeable. Now, in regards to patient education, this is such a huge space. You want to address their questions and concerns related to biosimilars. And as time moves forward, you will get more and more questions from your patients. You want to support your patients with complete and informed decision-making. In addition to the administration of biosimilars, the dose, strength, and route of administration will be the same as a reference product for any given indication. There may be slight differences in the delivery device and/or container, but again, the molecules are quite similar. In regards to the key messages, biologics cannot be made the same batch to batch. And by definition, biologics, including biosimilars, cannot be made generic. An approved biosimilar can be expected to have the same efficacy and safety profile as the reference product in the approved indications. Extrapolation is a new and novel concept and is justified based on the totality of evidence. Now, different patient considerations need to be taken. If a patient is doing well on a reference product, data from switching studies suggest that similar efficacy and safety can be expected with a switch to a biosimilar. However, patients who have developed antidrug antibodies to a given biologic should not be switched to another version of the same biologic, the originator or biosimilar. The availability of biosimilars has the potential to broaden patient access to biologic therapies and provide cost savings for patients.